Hello, good afternoon. Today's article that Leslie and I will be presenting are the sex differences in reward and punishment guided actions, brought to us by researchers at the Oregon Health and Science University. For many psychiatric illnesses, the prevalence and presentation is different for females and males. For example, major depressive disorder is more prevalent among women. Common symptoms among women are sleep disturbance, stress, and anorexia, while common symptoms for men include aggression, substance abuse, and risky behavior. This is just one example, but schizophrenia, ADHD, anxiety, and many more illnesses occur and appear differently among genders. How could this be? I'm sure you're wondering. Researchers Chowdhury, Wallenmiller, Rear, Park, Diaz, Simon, and Moghadam hypothesized that a neurobiological sex reason accounts for these differences. The goal of this research is to investigate the differences among sexes in three behavioral tasks that involve reward seeking and punishment. This relates to our research, as reward seeking and punishment avoidance behaviors are the basis for motivation. Ideally, researchers would be able to test their theories and so on and so forth on actual humans, but that is not always the case, especially when some sort of punishment is involved in the experiment. Nonetheless, animal models are great for understanding the behavioral neuroscience of sex differences, as they are not quite humans, but act like them in the sense of motivation. So they have similar behaviors of motivation as humans do. Much simpler, but similar. All right, so as I mentioned, rats were used for this experiment. They were randomly assigned and separated for the different experimental conditions as experiments with humans would have been. There were separate cohorts of rats for each task, and then the rats were 57 days old when they were delivered to the lab. They were either Long Evans rats or Sprague Dowley rats. Among the three tasks, a total of 50 rats were used. So there were three behavioral tasks, one being the punishment risk task, two being the approach avoid task, and the third being cognitive flexibility. I will go into deeper detail in these three in later slides. So they had to be acclimated to their surroundings, the chambers that they were in. So there were training periods for the first task. Those training periods, the data was not collected. Um, and then for the approach avoid task, that data was collected just to see how fast they were able to learn the task at hand, used for a basis, and then compared among genders. Um, but yeah, the training periods were also just to confirm that um, things were going as planned. Had they had anything go wrong, they would have used that time to adjust and do appropriate changes to further their experiment. All right, so task one, punishment risk task. There was a total of 12 rats used in this task, 6 female, 6 male, and these rats were actually the Long Evans rats. And They were 57 days old when they were delivered to the lab. So, as the title of this task implies, there was a risk of punishment after a certain task. Now, these rats were trained for 2 days on what exactly action they were required to do to receive a reward. And they were trained with no punishment for these 2 days. So they were trained to nose poke in their chamber to receive a sugar pellet. All right, so on our screen we have the diagram of this task. So first off, we start with the intertrial interval, and this was just a set of time that restarted the trials. There was an ITI after every trial before the next one began. Then there was a cue, and this was either tone, a light tone, or a light or a tone. Sorry, it's not the same thing. Two separate things, light or a tone, but it could have been together. And this just prompted the rat to take the action that they were trained for. And the action, as I mentioned before, was nose poking. So they would go to a little hole in their chamber, and as they were trained to do that, a pellet would drop and they would get their reward. But in this task, before the reward was delivered, there was 3 blocks, and in this block there was a probability of getting shocked. Now this was their punishment, and it was just a little shock to their foot, no worries. But there was 3 blocks, and on the screen you can see that block 1 had a probability of 0, block 2 had a probability of 6%, and block 3 had a probability of 10%. So for every block, the probability increased, and this is what they were looking to see. If their behavior or their action would have changed as the probability of getting shocked increased. So after the interval of possible shock, they were always given a reward, and they always took it. So there's the reward delivery and reward retrieval. The rats always made sure to get their sugar pellet, that's for sure. So the reward action was associated with an escalating probability of punishment. As I mentioned, there was always a reward after the proper action was taken, the proper action that they were trained for, but different blocks had different probabilities. Like I said, the punishment was just a brief shock to their feet, and the probability of the punishment increased from 0 to 6 to 10% in blocks 1, 2, and 3 separately. This task assesses how rats will behave to unpredictability and perception of potential threat. So please bear with me. This task was actually, there was a lot of moving parts, and is a little confusing, almost as confusing as this diagram without any information. So give me a second to explain everything, and it should all make sense by the end of this, I hope. So there are actually 4 main parts in this task. So we have the action for reward. So an action of either a nose poke or a lever press was performed for an immediate reward, that being the sugar pellet. The reward was always the sugar pellet. And then the second being action to avoid. So It was either a nose poke or the pressing of a lever to stop a foot shock. And then the third was cued action for reward. So the cue that I mentioned earlier of either the light or the sound tone was initiated, which prompted the rat to nose poke or press a lever once it became available. Now, this cue only lasted for 10 seconds, and the action must have been performed during the cue to get a reward. And the fourth being cue action to avoid. So, same 10-second cue that must be performed during, same 10-second cue of a toner light, and the action of nose poking or lever pressing had to be done during this time. And they were again avoiding a shock to their foot. So, on to task 2, the approach avoid task. So, 22 rats were used in total, 11 female and 11 male. These rats were actually the sprawl dowley type of rats. And these ones were also 57 days old when delivered to the lab. All right, so that being said, if we're looking at the diagram, there's the ITI for every trial, and either a reward cue or an avoid cue occurred. I'll just describe the reward cue first, and then we'll go to the avoid cue. So with the reward cue, if they perform the action that they were trained to perform within the 10 seconds of the cue, then this indicated correct action and they were rewarded. So there was the delivery of the reward and then the retrieval of the reward, and then it circled back to the ITI, intertrial interval. Had they not performed the action in the allotted time, then they would have not received anything and they would have just gone back to the ITI. Now for the avoid cue, if they did the trained action within the 10 seconds, then they were not shocked. So it would go straight back to the ITI. But if they did not do the correct action that they were trained for within the 10 seconds of the cue, then they were shocked. So here I would like to mention that the rats were actually given the drug FG7142 after doing these trials one time. They were then given the drug and they were later assessed. This drug was used to ease anxiety, and they measured their anxiety levels in the different tasks after administering the drug. FG7142 was dissolved in 0.09% saline with 15% VV tween 80 for a 1 milligram kilogram injection. There were 4 doses, one every 7 days, and yeah. Task 3, cognitive flexibility. Now this task was really assessing what the name implies, how flexible their cognitive abilities were. And you'll see why after I further explain. So this task had a total of 16 rats that were used. There were 8 female, 8 male, and these were Long Evans rats. So there was a 9-day training period. In this training period, they were initially trained to nose poke and then nose poke in less than 10 seconds. And then they were administered a side bias test. This side bias test just assessed which side of this chamber right here that they would initially go to. In the side bias test, 2 nose ports were available on either side of the chamber. The rat could go to either to receive a sugar pellet. On the following trial, the rat was only given a sugar pellet if they nose poked the opposite port. This was repeated 7 times, and whichever port they went to first 4 more times was considered the side that they were biased to. Now there was a set, shifting, and reversal learning task. So a cue of a light would have been initiated in one of the nose ports, prompting the rat to go to it. If they went to the port within the allotted time, they were rewarded. And if they did not go within the allotted time or at all, the trial was reverted to the inner trial interval, waiting for the next trial. The illumination of this cue light happened in pairs, so pairs of trials. So if on the first trial it happened on the left nose port, the following would happen on the right. But if on the third trial, it could happen on the right and the fourth would be on the left. Yes. So this was a random order, but it just tried to get get the rat to understand that the cue meant that there was food on that side. The initial discrimination task was when rats were assigned to the nose port opposite of their initial side bias. They were meant to ignore the stimulus of the light cue and respond to their assigned side. Once they proved that they had learned this task, it was reversed so that the rats had to now respond to the opposite side. And once they proved that they could do that, the stimulus of the light cue that I had just mentioned was now put into the task. So now they actually had to respond to the light, which before they had associated as not important and irrelevant. So for the initial discrimination, reversal, and set shift tasks that I explained, um, they were allowed to move on to the next ones um, among those 3 once they completed 8 consecutive trials. So that's what I meant by proving that they learned it, that they learned the previous task. Had they not been able to make a successful 8 consecutive trials, then they were just moved on to the next task the following day. Yeah, I probably would have been one of those rats that would have just had to been moved on. Okay, finally the results. So, for task 1, the punishment risk task, as you can see in figure B in the lower left of the screen, we have the 3 blocks on the x-axis, and each block had 30 trials in them. And then on the y-axis, we had times. So this graph shows the cue response latency, so how long it took the rats to respond to the cue. So that's why we have time on the y-axis, and the block as let me remind you that the blocks had a probability of shock being 0 on 1, 6% on block 2, and 10% on block 3. So they were seeing if the increase in probability of punishment increased the response cue, and results showed that there was a significant effect in the increased cue response latency as the probability of punishment increased. Sorry, that was a mouthful. These results these results suggest females were more affected by the risk of punishment. So as we see in block 3 with the highest probability of punishment, the blue data points indicating female, while the orange indicated male. The blue data points are much higher in the time, so they took a lot longer to respond to the cues as the probability of punishment increased. However, if we look at figure C, we see that the blue and orange data points are pretty close. This revealed that there was no significant difference in reward retrieval latency. Let me explain that. So the reward retrieval latency was how long it took them to retrieve the reward. And one thing about these rats, they always went for their reward. They were not playing around with their food. These results suggest that both male and females have similar levels of motivation, as the sugar pellets were their motivation, because rats are highly food motivated. So the fact that there was no significant difference in the reward retrieval latency shows that there was no significant difference in their levels of motivation. So task 2, approach avoid task results. We are looking at the reward training. So let me remind you, the reward training is they did an action and they were trained to do an action and they were rewarded for it. So on figure B, we have the cued reward as a function of days to criterion, meaning the amount of days it took the rats to meet the criteria, as a function of their gender. So females in blue and males in orange. If we just look at figure B, we can tell that there's no significant difference, and a statistical analysis confirms that there is no significant difference in the reward acquisition among females and males. Okay, so I know figures C and D look like a hot mess, but there is a method to this madness. It's actually going deeper into what figure B was trying to show. Now, figure C and D are showing the same thing, but in regards to female and male separately. So C being the females cued reward training and D being the males cued reward training. So as I mentioned for figure B, there was days to criterion on the y-axis. And by criterion, I mean a 70% successful rate. So that meant that 35 out of 50 trials were performed correctly, meaning the action was correctly done by the rat. So on figures C and D, on the y-axis, we have number of correct trials out of 50. And then on the y-axis, the day of training. So as you can see, for both graphs, day of training closer to 1 had much, much, much lower number of correct trials as opposed to the data points closer to day 5. Yes. Just to reiterate, although graphs C and D look like there would be a significant difference, there actually was not. C is just a little bit all over the place, but all in all, there was no significant difference in the reward acquisition among females and males. So this indicates, or these results suggest that they both learned at the same rates, and there's nothing significant about one gender over the other. So the last 3 graphs were on the reward training, and these 3 graphs are on the avoidance training. So they were set up the same exact way as I had just described for the reward training, but these data points are on the cued avoidance training of the female and male rats. The overall result remains the same. There was no significant differences in the avoidance acquisition among females and males. On figure E, we see similar trend among females and males in the days to criterion, which again was a 70% successful rate, meaning that the rats did the task to avoid the shock. Yep. Now these results are show the actual task performance with the reward trials being on the left and the avoid trials being on the right. The x-axis for all 4 graphs are the days prior to the FG714 test, which was a drug test. So it's these graphs are showing the performance of the task over the 5 days before the drug treatment. And so on the y-axis, we have the proportion correct. So how many times the female or male performed the reward trials correctly, or the female and males avoided or performed the avoidance trials correctly. If we look at figure H and I, the reward trials, females being H, I being males, we see that they're pretty similar, and the statistical analysis confirms that there was no significant difference in the reward trials among females and males. On the other hand, if we look at J and F, we can see that the female avoid trials graph J is a lot more sporadic than graph K. And a statistical analysis confirms that there is a significant difference in the avoidance trial among females and males. Females failed to avoid the shock more often than males failed to avoid it.
In these graphs, researchers reported the proportion of correct responses at the varying doses of the drug FG7142. So, again, this drug is an anxiety producing drug, so they wanted to know how it would affect their performance on these tasks. For both of these figures, the x-axis is the different doses of the drug with increasing doses as you move to the right, and then the y-axis represents the proportion of trials they got correct. The blue shapes are females and the orange are males. They used 11 female rats and 7 male rats. So in figure A to the left, this represents the number of correct reward trials, so how often they did the nose poke to receive a pellet of food. And we saw that the drug affected both sexes equally, so both showed less correct responses with higher doses of the drug. And then in figure D to the right, this graph is a measure of the times the rats correctly avoided the shock. So the avoidance behavior was diminished with the drug, so they all correctly avoided the shock no matter the drug dose. But males were more sensitive to the drug, meaning they correctly avoided the shock less times with higher drug doses. In these graphs, researchers reported the latency of the rats in the various tasks. So for these graphs, the x-axis is still the increasing doses of the drug, but the y-axis is in time since they're measuring latency or the delay in the rat's reaction time. So starting on the left with figure B, they found that higher doses of the drug showed increased latency. So the rats took longer to respond to the light or sound, whatever they used as the cue to get the food pellet. However, there were no differences between the male and female rats, so they both responded the same to the drug. And then moving to the right onto figure C, they measured how long it took rats to get the food pellet by itself without any cue, so no light or sound. And the rats showed no hesitation in obtaining the pellet for the most part. Moving to figure E. So this graph looks at the response time in getting the food pellet when the light or sound, the cue, was followed by a shock. So rats showed increased latency or more hesitation with higher doses of the drug. Females also showed a longer latency period in responding to the avoidance cue. And then in figure F, the one all the way to the right, they also showed the times that they failed to avoid the shock, but eventually escaped it. There were no main effects, and most of the rats were quick to escape the shock. But this is probably because it was an uncomfortable feeling, so they were quick to escape it. These are the results from the EPM, the maze that they made the rats do, which measures anxiety. So they measured this by the time spent in each arm. So time is on the y-axis, and then the open versus closed arm is on the x-axis, with females in blue and males in orange. So, starting with figure G to the left, they found no sex difference, so each sex spent equal time in each of the arms. And then in figure H, we see the time the rats spent in each arm after performing the approach avoid task. So they found that females spent more time than males in closed arms and less in open arms after the task. And then in figure I, this graph just shows the flinch response to the shock. So we see that there's no significant difference between the males and females. They showed relatively the same response to different intensities of a shock. From the cognitive flexibility task, they found that all the rats, regardless of sex, required more trials to complete the reversal learning task. So, this is a task where they had to poke the opposite side as the one that lit up in order to get the food pellet. And this is what's shown in figure B with the number of trials it took them to reach the criterion on the y axis and each of the three tasks on the x axis. And then figure C shows the number of premature nose pokes the rats made, with the number in each session on the y axis, and then again each of the three tasks on the x axis. So from this, they found that males had more premature nose pokes than females. So, from this study, we can conclude that there are no differences in learning or executing actions between the sexes, but we did see that females learn to avoid punishment faster, and this was found by Dalla and Shores. We also learned that aversive behaviors were context dependent in females. So, females were more sensitive to punishment if it was uncertain, but they were less sensitive if they were certain they could avoid punishment. There were no differences in reward motivated behaviors, so both sexes were quick to respond when only a reward was involved, no risk of punishment. Sex differences were revealed when the threat of a punishment was introduced. So, like I said previously, females were more sensitive to uncertain threat, and a study done by Orsini and colleagues found that female rats are more likely to avoid rewards that might be associated with threatening stimuli. So, something like a shock or another type of punishment. This may be responsible for the increased vulnerability to anxiety seen in women. So, they become more anxious when they're forced to make these decisions that could result in a punishment. This also indicated that males were less responsive to a potential threat, and this reduced responsiveness to risk in males is consistent with higher incidence of impulsivity, and this was found in a study done by Perry and Carroll. Researchers also found sex differences in performance on the EPM, the maze that measured anxiety. So, females showed greater anxiety like behavior, and this indicates that they may be more resilient than males to aversive events, but continued exposure to these aversive events may lead to increased anxiety. And this is consistent with a study done by Martin and colleagues. So, this suggests that women's vulnerability to anxiety stems from these accumulating aversive experiences. They also found that there was no sex difference in task performance, but they did find that males made more premature responses. And a lot of the psychiatric disorders that preferentially affect males are associated with this behavioral disinhibition, which is a pattern of antisocial, impulsive, and sensation seeking behaviors. So, this indicates that differences in neural circuitry involved in these behaviors could predispose males to disorders, and this is supported by many studies done. Also, increased impulsive action increases risk for drug abuse and addiction, which could explain the link between these disorders and the impulsive behaviors seen in males. There was also differences found in the strategies to assess punishment risk and avoidance behaviors. In females, it was highly dependent on the context, and we saw a split between the females who avoided the shock versus those who escaped the shock. So these females who are escaping the shock are both confirming the shock still exists and that the escape method still works, which may be facilitating behavioral adaptation, and these escapers may be showing priorities of self survival and maternal behaviors. And finally, the researchers found no difference in anxiety levels on the EPM despite the well known sex differences in these anxiety related psychiatric illnesses. So, this shows that EPM is an invalid measure of anxiety processes that are relevant to differences in sex in regard to mental disorders. So, from the study, there's other aspects that can be investigated in regards to sex differences and psychiatric illnesses. The first would be looking at the mechanisms underlying these differences. So, looking at the molecular basis of these disorders in males versus females. Another thing that could be looked at is the group of avoiders and why these behaviors were prevalent in some females, and also why they weren't observed in males. Eventually, this could be done using a human model where people of the two sexes complete tasks that measure anxiety, but this is complicated because of the ethical issues that arise from inducing anxiety in participants. Thank you, everyone, for listening to mine and Daniela's presentation. Daniela can't be here, but I'll be around to answer any questions that anyone may have.